Adolescence is defined by a bizarre contradiction: cognitive peaks mapped directly alongside behavioral cliffs. Put a teenager in a controlled laboratory setting, and their logical reasoning matches that of adults. They can calculate probability, absorb information, and assess risk with equivalent mature competence. Yet epidemiological data from the exact same age bracket shows a massive spike in behavior-driven preventable mortality. Motor vehicle crashes, substance binges, and risky decisions surge despite their intelligence. This is the adolescent paradox. We are left to reconcile high cognitive competence with a sudden onset of catastrophic decision-making. These conflicting datasets suggest that for the adolescent, cognitive competence and behavioral control are independent variables. The discrepancy originates deep inside the frontostriatal circuitry, a physical mismatch in how different regions of the brain mature. The standard neurobiological explanation is the dual process model. It posits a biological conflict between two areas: the subcortical reward system, which drives emotion and instinct, and the cortical control system, which handles rational analysis. But this biological dual system only accounts for the potential for risk. It fails to explain the variance within a single individual: why a teenager makes a highly dangerous choice on a Friday night, but a cautious, logical one on Saturday morning. Traditional models evaluate adolescent decision-making in a vacuum. They ignore the specific external environments where these decisions actually happen. To predict actual behavior, we must move beyond the dual system and construct a tripartite model, one that integrates environmental context as an active, multiplying variable. The first component of this system is the brain's subcortical engine. It runs on dopamine, originating deep in the midbrain. This chart maps the specific dopaminergic pathways. Axons project directly from the substantia nigra and the ventral tegmental area, pushing upward into the striatum. Notice the pathways terminating in the nucleus accumbens. This highly specialized region processes novel, rewarding stimuli. During adolescence, this system displays unique dynamics. The basal, or resting, rate of dopamine release is lower than in adulthood. However, when the system is stimulated by a reward, the dopamine release is significantly larger and persists much longer. These mechanics sparked a debate in neuroimaging. Does a low baseline mean adolescents are reward deficient, requiring extreme risks to feel anything? Or are they hyper-responsive to the rewards they do encounter? Converging fMRI evidence settles the argument. The adolescent ventral striatum is hyper-responsive. It reacts to potential rewards with disproportionately massive surges of neural activity. Once stimulated, this intense dopamine rush creates a potent reinforcing feedback loop. The brain registers a massive reward signal and immediately motivates the individual to seek more. The adolescent striatum operates as a hypersensitive engine, primed to translate motivational drives into immediate physical action. Now consider the physical brakes. The frontal lobe is responsible for higher cognition, risk evaluation, and inhibitory control. A critical hub here is the orbitofrontal cortex. It receives dense dopaminergic input from the midbrain, but it undergoes a highly protracted, delayed maturation process. Researchers use functional MRI during go-no-go cognitive tests to measure how efficiently this control system operates across different age groups. The scans reveal a stark physical difference. Adults display tight, concentrated, high-intensity neural recruitment. Adolescents exhibit a diffuse, broad wash of low-intensity activation across the frontal lobe. This diffuse activation pattern is the signature of an immature inhibitory control system. It simply works less efficiently than in adults. The synaptic pruning required to fine-tune these prefrontal projections will extend well past the teenage years, taking a decade or more to finalize. The biological setup is clear. The reward engine is revved up and acutely sensitive, while the cortical brakes are still under construction. Mapping these two diverging timelines together gives us the formal imbalance model. If you plot functional brain development over a human lifespan, the subcortical affective networks follow a steep, nonlinear arc. They peak aggressively during adolescence. Conversely, the cognitive control networks, the prefrontal cortex, follow a slow, steady, and strictly linear maturation. Look at the intersection during the teenage years. A massive vertical gap opens up between the high limbic peak and the still developing cortical line. This is the literal zone of imbalance. This structural gap provides the biological basis for nonlinear shifts in behavior. It heavily favors intense affective responses over analytical restraint. During this exact window, the individual is biologically biased by their functionally mature limbic regions, overpowering their own delayed prefrontal control. Yet, as physically accurate as the imbalance model is, it remains incomplete. Analyzing a brain in a scanner strips away the socially dense reality of being a teenager. We have to introduce environmental context, specifically the presence of peers, as the third multiplying variable that actually dictates behavior. A study by Chain and colleagues tested this directly by placing adolescents in a simulated driving game. They measured striatal responses during solo play and compared them to play while observed by peers. The results showed that peers operate as a conditioned stimulus. They are a highly salient social reward that directly triggers the adolescent dopamine system. The mere presence of peers in the room caused an exaggerated spike in ventral striatal activity. This subcortical surge triggered a secondary effect, a measurable decrease in prefrontal cortical engagement. Social context reconfigures the brain's internal math, tipping the already fragile frontostriatal balance toward the immediate reward. This brings us to the tripartite model: the striatum, the prefrontal cortex, and environmental context. In a cold environment, the developing prefrontal cortex functions well, utilizing logic to avoid danger. But in a hot, socially salient environment, the context node activates as a multiplier. It feeds directly into the reward system, expanding the striatal response and dimming prefrontal controls. This physical neural override drives the adolescent toward immediate reward, even when they possess full cognitive awareness of the mathematical risks involved. Adolescent risk-taking is a context-dependent neural override. That is how high intelligence pairs seamlessly with catastrophic choices. Understanding this physiological reality challenges how society approaches teenage safety. Interventions typically rely on educating teenagers about the statistical dangers of risky behavior. Education alone fails because adolescents already comprehend the risks. Their prefrontal cortex understands the data perfectly well. Clinical treatments and public policies must pivot. Instead of exclusively treating the individual's logic, we must modify the external context. Interventions must aim to mitigate the specific social conditioning and external triggers that actively exploit this temporary biological imbalance. To effectively protect adolescent lives, we have to stop treating their intellect and start treating their environment.